right, welcome to Collaborative Sciences. I can't remember the title, sorry guys. I hope you had a great evening last night. Here are some housekeeping notes for you guys. Please silence your phones. Remote comments will be muted upon joining online sessions. Photography and video recordings of sessions are prohibited. APR's social media policy is that everything can be shared, and the speaker asks that it not be shared. Please respect the speaker and do not post any social media. Hashtag for the event is hashtag APRF 2023. Remote participants may post questions in the online chat. In-person attendees may ask questions at microphones in the room. They're on every wall, the tables in the center. Download the APRF meeting app to access poster and session abstracts, connect with other attendees and sponsors, and create your personalized meeting schedule. Explore the app to find exclusive extra content. Remember to visit the ABRF exhibit hall to engage with ABRF sponsors. The exhibit hall hours are today, 9 to 4:30, tomorrow, 9 to 4:30, and Wednesday, 10 to 4. And please visit the ABRF posters, which will vary daily, featuring research and information posters on a range of technologies for full facilities. A gallery of electronic versions of all posters is available at the annual meeting website and through the app. The 2023 Technology Showcases feature leading partners who will review the latest instrumentation and software solutions. The showcases take place today from 7:30 to 8:30 and 6 to 7, tomorrow 7:30 to 8:30, 6 to 7, and Wednesday from 7:30 to 8:30 a.m. And session evaluations will be available in the app. In addition, all attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. One respondent will be selected for a complimentary registration for 2024 annual meeting. The survey also will be accessible through a QR code displayed on site and posted in the event app and attendee hub. Questions, concerns? Our sponsor for this session is Roche Boshin, and he will come up and do his thing.
Thank you.
Thank you.
Good morning, everyone. Thank you for joining us today for a discussion on digital PCR. My name is Boshin Gao, and I'm with Roche Sequencing and Life Science. So today I want to talk a little bit about what is digital PCR, some key advantages of digital PCR over traditional qPCR, and some of the applications that can take advantage of these benefits. And lastly, I want to briefly introduce the newly launched Roche digital PCR platform called the Roche Digital Lifecycle. So what is digital PCR? I'm sure everyone here is very knowledgeable about qPCR, but digital PCR is this new application that's based on PCR with an important concept of partitioning. So essentially, you start with a prepared PCR reaction with your usual master mix, primer, probes, and sample. And instead of running this reaction in one single tube, you will partition this to 10,000 or 100,000 smaller micro reactions. And then these smaller reactions will go through thermocycling, and the endpoint signal is collected and counted for positive versus negative counts. And then finally, a Poisson calculation is applied to calculate the concentration based on these positive negative read and account for the reaction volumes. So the key advantage of this application is that it offers true absolute quantification. In a regular qPCR, you compare all of the samples to a standard, but in a digital PCR, it allows for true absolute quantification without the need to compare to a reference standard. This allows increased detection sensitivity for especially for low abundance target due to an enrichment effect. So on the left here, we have a light blue mutant target that's only 1/8 the concentration of the wild type. And in a conventional PCR, it might not amplify just due to competition. However, by partitioning down and dividing this into smaller micro reactions, the small mutants can, the proportion favors the low abundance target, and it can be picked up. And on the right here, it shows the digital PCR delivers a high precision, here shown as low CV confidence level over a wide dynamic range. And another key advantage is that with increased number of partitions, the precision is increased. So there's always a place for qPCR, and Roche is proud to offer a wide range of products that support it. But I just want to highlight some of the additional advantages of digital PCR so that they may be of interest to your research or projects. Here are the 5 main advantages I want to highlight. Absolute quantification, high precision, high sensitivity, reduced competition, and increased tolerance to inhibitors. So first, absolute quantification. In a traditional qPCR, you're comparing all your results to a standard. So a standard is required, and you are impacted by amplification efficiency, and you're looking at a CT value that can be varied from plate to plate, from instrument to instrument. With digital PCR, there's no standards. You're only looking at the endpoint fluorescent signal, and it's independent on PCR efficiency. And at the end, you get a result of positive versus negative, so essentially a yes or no. And the second advantage is high precision. So qPCR, your results is affected by the quality of your standards, the CT reading from instrument or from runs, the reaction volume, and instrument variability. With the digital PCR, once you choose your configuration, your partitioning configuration, you're always running the same volume per reaction, and you also have the same number of partition per run. So this takes a lot of the variance out of the PCR. And at the data interpretation stage, again, you're looking at endpoint data, so it's just concerned with positive or negative, and there's no reliance on standards or CT value. The third advantage is increased sensitivity. So here an example, if you have a 20 microliter reaction with 2 copies of target in there, your concentration is effectively 0.1 copy per microliter. However, with partitioning down, it will effectively increase the effective concentration by subdividing into 10 partitions. Each reaction, the concentration is brought up to 1 copy per microliter. So this is the enrichment effect. The enrichment effect also leads to advantage of reduced competition. Like mentioned in the previous slide, with low abundance targets, this enrichment effect will allow the target to be more favorable, and it does not need to compete as heavily for PCR reagents. So it allows for increased detection of this low abundant target. And lastly, by partitioning down the reaction, it reduces the effect of inhibitor. So in the qPCR, the CT value is affected by inhibitors such as enzymes, primers, probes, buffer, or standards. However, in the digital PCR, because only endpoint signals are being considered, there's no reliance on the reaction kinetic. And by partitioning down these reactions, the individual interaction with inhibitor is reduced. So keeping in mind of these advantages of digital PCR, here are some of the applications that we think is more beneficial when digital PCR over traditional qPCR. So there's the copy number variation, CNV analysis, where resolution is very important. So using a large number of partitioning is more favorable for this application. On the other end of the spectrum, for rare mutation detection, you can take advantage of digital PCR's increased sensitivity, lineage and phasing analysis, RNA detection, and detection and quantification of microbes and viruses, where the inhibitor is a big challenge. So the increased tolerance to inhibitor can help with these applications. And lastly, any application where the absolute quantification is important, you can take advantage of digital PCR. So for final part, I want to briefly introduce you to the new Roche digital life cycler system. The digital life cycler system features a number of technical advancements that together make it a very powerful clinical research tool. So the power lies in the three number, 365. We have three different nanowell plate configurations that ranges from 20,000 partition, 28,000 partition, and 100,000 partition. So you can fine-tune your research based on your application if you want higher resolution or higher sensitivity. And these nanowell design increases the efficiency of partitioning will reduce the chance of reaction contamination. And for 6, we have 6 advanced optical channels. So this allows a high degree of multiplex for both DNA and RNA application. And lastly, we have a 5x concentrated DNA and RNA reaction master. So this mix allows for greater sample input by providing a 5x concentration reagent. Okay, thank you for your time, and if you have any questions on digital PCR application or on the Roche digital life cycler system, please come by our booth at number 400. Thank you again for your time today.
One of the pleasures of working in a core, such as the one at MIT, is that I get to work with people like Dan and Adam. They're really fun to work with because you ask questions, they answer. Cool, right? Dan is one of our radiation oncologists who works at Israel Deaconess, and he works here at MIT as well, the Matt Vanderheiden lab. And then Adam, I want to do some of course, but we work with the entire data scientist. So it's a great group of people to work with. As I'm sure you guys know, core people are pretty awesome. So thank you, Dan, for your
Thank you, Kathy, and thanks to the AERF meeting organizers for the invitation to talk today. So this talk is going to be maybe a little different from some of the other talks that you're hearing at this meeting. But I am, as Kathy mentioned, a radiation oncologist at the IBMC and also a cancer biologist. So what I'd like to do today is talk about some preclinical cancer studies that we've been doing in the lab and really highlight how the core facilities at our institutions were critical in helping us to get this study done. We generate, of course, lots of data by doing these studies, and so the second part of the talk, Adam Kacha, data scientists in our imaging core facility, is going to talk to you about some potential ways to maximize the use of that data. So jumping into the topic of cancer, every year we make breakthrough discoveries in cancer research, but really not that many of them make it to the clinic, unfortunately. And so a major bottleneck, in my opinion, is the translational component, of which preclinical studies are a key component. So I would argue that mouse models are an excellent space in which to conduct preclinical studies because one can simultaneously look at efficacy of a treatment, but also toxicity. And that's really important when considering a novel cancer therapy. And so today what I'm going to do is I'm going to walk you through an example of how in the lab we used a mouse model to identify a novel target in prostate cancer. And then I'm going to talk to you about how we ultimately designed a preclinical trial to test that. So prostate cancer is the second leading cause of cancer death in the United States. And PTEN is a tumor suppressor well established in many cancers, but is very frequently mutated in prostate cancer. In advanced prostate cancer is about 50% of cases. It's with poor prognosis, which means that the current treatments that we have are insufficient. So we need better treatments for this form of prostate cancer. It may not surprise you, sorry, lagging behind one slide. It may not surprise you, given what I've just told you, that if you delete PTEN by itself in the murine prostate, you can actually develop cancer. So this is an experimental model in the mouse where de novo or spontaneous generation of prostate cancer is virtually unheard of. By just deleting this tumor suppressor, you can induce cancer. And so we've been using that model to try to figure out better treatments. The PTEN is a tumor suppressor. It's involved in the PI3 kinase pathway, which is an important pathway for regulating metabolism. So that's one of the first things that we looked at, is we looked at how metabolism is different in these tumors. And what you can see here is a collaboration with our imaging core facility. This is a PET study that was done showing that you have increased glucose uptake, specifically within the prostates of these tumors when PTEN is deleted. The other thing that we found is that pyruvate kinase, which is the last enzyme in the glycolytic pathway, a specific isoform of pyruvate kinase is overexpressed in these tumors. So shown here, a nice histology panel showing expression of two isoforms of pyruvate kinase. PKM1 is a high-activity isoform. It's almost not expressed in prostate cancer. And PKM2, which you can see here, is strongly expressed and expression increase as we progress through prostate cancer. So glucose uptake is increased. The terminal enzyme, the specific isoform, is overexpressed in this cancer. So what happens if we take away pyruvate kinase, the N2 isoform? So that's shown here on this slide. This is a specific deletion of the N2 isoform of pyruvate kinase in the context of PTEN deletion in the mouse prostate. And it turns out what these tumors do is they switch from PKM, which is what we delete, PKM2, which is what we deleted, to PKM1. So they switch to the other isoform. Now, one thing I'm not going to go over today that's important is that the PKM2 isoform is the isoform of pyruvate kinase that's overexpressed in virtually every cancer cell line that we use in research. And so it's important for proliferation. And what you can see is that when you switch from PKM2 to PKM1, this causes a profound suppression of prostate cancer development. So that's what you can see on the panel there on the right. So knowing that PKM2 is involved in glycolysis, helps to regulate the glycolytic pathway, we collaborated with the metabolomics core facility at the Whitehead Institute, part of MIT, just across the street from us, to try to understand if we could gain some mechanistic insights into why we're seeing tumor suppression when we switch from PKM to PKM1. And so we profile about 150 different metabolites. In the panel on the left, what you can see is that the tumors, when you switch from PKM2 to PKM1, have decreased glucose uptake, almost virtually the same as a normal prostate. And on the right, this is a principal component analysis, which summarizes the results of about 150 different metabolites. And what you can see in this principal component analysis is that the tumors that switch from PKM2 to PKM1, those are the orange ones, they take up a very different metabolic state. So when we looked a little bit more deeply at which metabolites are altered, what we found was shown on this slide here. On the right, you can see that there's a significant reduction in nucleotides, so specifically purine nucleotides and their precursors. On the left, you can see that there's an increase in nucleosides. An increase in nucleosides, which suggests that there was increased nucleoside turnover. And so this really suggested to us that there was something going on in the nucleotide biosynthesis pathway in these tumors, causing their suppression. And what we found ultimately is that there was a switch in PKM isoform, that the switch in PKM isoform expression resulted in replication stress, as you can see based on the phospho-CHEK1 immunohistochemistry panels on the left, and a promotion of senescence, shown by the beta-glycosidase staining in the panels on the right. Again, I'm going to highlight here, couldn't have been done without the help of the core facilities and helping us to get these studies done. So the model that we end up with is shown here. P10 loss in the prostate induces adenocarcinoma, which in the presence of PKM2, the low activity form of pyruvate kinase, results in tumor progression. However, when we genetically force the tumors to express instead PKM1, this results in tumor suppression and ultimately replication stress and senescence. So that's great. We have a nice genetic pathway here that suggests that that pyruvate kinase is important in prostate cancer. What we need now is a therapy, right? So it turns out that pyruvate kinase activators have been developed for patients that have pyruvate kinase deficiency in their blood. So these are pediatric patients that lack pyruvate kinase or have low activity pyruvate kinase in their red blood cells. And so it's a drug that's in clinical trials. It's safe even in the pediatric population. So we asked, well, what happens if we treat these tumors with a drug that activates pyruvate kinase, essentially making PKM2 isoform act like PKM1? Those results are shown here. Apologies for the formatting, I think you can still see it. On the left, there is a waterfall plot which shows tumor volume changes in animals that have been treated with vehicle or with a drug that activates pyruvate kinase called TEC46. And I think it's pretty obvious here that the tumor progression is slow, and some tumors actually regress. Panels on the right, again, in collaboration with our imaging core facility, we were able to show, in this particular example, you can see actual regression in tumor volume after being on the drug for just 30 days. So top panels in axial and coronal section of the prostate before treatment, 30 days later, the bottom two panels, you can see the tumors have gotten smaller. I have to highlight, of course, our histology core as well. So these tumors do not surprisingly also have a lower proliferation rate. This is a Ki67 immunohistochemical staining, which is a marker of proliferation. You can see the tumors have significantly less proliferation. So we have a drug now that seems to be effective in this mouse model. What next? What I told you earlier, this is where a lot of drugs fail, is that we have these discoveries, but then they never make it into the clinic. So as a radiation oncologist, I can tell you that, again, sorry for the formatting issues here, I'll walk you through it, that patients with high-risk prostate cancer is a population where we need to do better in the clinic. So these patients are typically treated with surgery or radiation therapy plus androgen deprivation, both equally as effective, but only are effective in 50% of cases. So 50% of men that are treated according to the standard of care will recur within 5 years. And there's really nothing adjuvantly that we offer them. So they get the treatment, and then we wait. We follow the PSA, and we wait until they recur, and then they have to undergo additional treatment, salvage therapies, energy deprivation, all of which have a fair bit of toxicity. And so the question that we asked is, where could our drug be useful? So one option would be in combination with radiation therapy, or energy deprivation, potentially with surgery, potentially in the adjuvant setting. So after they undergo the treatment, we now have a drug that's benign in terms of side effect profile, but they go on this drug afterwards and prevent progression. So we wanted to design a clinical trial to test this. Now it turns out that doing radiation therapy is not that easy for us. So we had to develop a platform, and the platform had to be something that we could use at our institution. So I'll quickly walk you through these slides here because I think it's an interesting sort of quick story. So this is a research radiator. Those of you who work in animal facilities probably know these. It's got 2 gamma cell sources in universally irradiated mouse. It's typically used to ablate the bone marrow. So we said, well, can we adapt this research radiator to focally treat the prostate? So I didn't mention this, but when we treat prostate cancer with radiation, we're giving multiple fractions of radiation per day in order to give you a dose that's curative. So we needed to we wanted to simulate that in the lab. So we were able to develop a set of lead shields that collimate down the radiation beam. And then the second challenge was, how do we then target the prostate? So we've got a radiation beam that can look like the radiation beam we use in the clinic. But how how do you mobilize and target prostate? And that was actually not a trivial question. So if you look at the CT scan here on the left, you can't make out what prostate is. So we overlaid this with an MRI. With the MRI, you begin to see what prostate is, but still, to the untrained eye, it's really challenging. I look at MRIs all the time. I don't look at mouse MRIs, but it's for someone who looks at MRIs, even there, it's challenging. So we collaborated with the Histology core facility to integrate some histologic data to actually identify where the location is of prostate in the mouse, and then be able to immobilize that so that we could treat it. They're shown on the bottom right panel there is a immobilizer that we designed specifically to immobilize and treat the prostate. So it works. The immobilizer and the target prostate shown here is an example of a mouse that's been set up in this setup and CT image on 2 separate days. And you can see in the overlay there, the bony anatomy that the setup alignment is really excellent. So this now allows us to deliver fractionated radiation therapy like we deliver it in the clinic to the mouse prostate. So verification that the platform is working is shown here. So gamma H2AX is a marker of DNA damage. It's a immunohistochemical marker. The panel on the left, you can see an untreated prostate. On the right, this is the treated prostate. Universally throughout the prostate, you can see an induction of DNA damage throughout the nuclei of the prostate cells. So our platform works. Another nice example of this, if you come by the Coke facility, you'll see this hanging in our gallery. So this image was selected as an image board. So kudos to our histology for people sitting there in the back. So this is the mouse prostate. So the mouse colon. The colon sits right behind the prostate. This is a 2 centimeter radiation field. And what you can see is that DNA damage shown by the pink cells there is induced only within the cells that are within the radiation field. So a nice example of how we can focally deliver radiation to the prostate. So shown here is a study that we did showing that the treatment works. So this is a 5-fraction radiation regimen. Mice get radiation on 5 consecutive days. This is we use it as a proof of concept. We use a xenograft mouse model. So these mice have got tumors implanted in both flanks, and we treated only one. What you can see in the example here is that the treated tumor regresses completely, while the one on the opposite side, the shielded side, grows. So confirming by pathology, we show that we can achieve pathologic complete responses using this platform. Tumor cells completely gone, and some percentage of these mice. So the clinical trial that we decided to set up was testing PK activator in combination with radiation therapy. And we wanted to benchmark also against energy deprivation. So the study that I'm just going to briefly describe for you, and I'm going to turn it over to Adam, is the one shown here. This is a complicated diagram. I know you can't read all of it, so I'll just walk you through some of the highlights, which Adam's going to then talk about how we begin to think about analyzing this data. We start out here on the left. We start out on the left side with a P10 mouse model. We then take these animals, we do a baseline MRI just to see what size their tumor is, and then we stratify them based on the baseline MRI. So these animals are somewhere between 13 and 14 months of age at the time that they enrolled on the study. Once they get the baseline MRI, then we randomly allocate them to one of 3 different arms we have here. We have vehicle control, we have our novel drug at 46, and we have enzalutamide, an androgen deprivation treatment that we use in clinic. The animals are on that treatment for about a week, and then we randomize them again to radiation or no radiation, combination therapy. So it's getting pretty complicated here, but the goal was to identify how to best use the drug in the clinic, so that's why we felt that this was an essential component. So they were randomized, and ultimately then you got 6 different groups. So then we get another MRI. Remember, these are prostate tumors. These are not xenografts where you can measure them. So you need to do imaging in order to track the tumor size. We got another MRI just at the end of radiation therapy, and then we waited another month out to get that 3rd MRI. A lot of data was being generated. Then we followed these mice until death. So that was the ultimate end point of the study, was to see how these treatments are affecting survival. So the results of the study, these preliminary data, we saw a few mice that are hanging out in the animal facility, are showing that there were some surprises. So the first surprise to me as a radiation oncologist is this model is very radiation resistant. So I was not improving survival by treating these mice with radiation, which is important. And so that's something that we're looking at now, is the role of P10 in inducing potential radioresistance. The positive controls of enzalutamide did work. You can see that here. And our drug, importantly, 30 days of treatment with the drug did not improve radio sensitivity. That's something we're looking at now. So it might be that we didn't treat for long enough, but this is where all the work is now, is looking at the imaging studies. So what happened to actually tumor size after the end of radiation, 30 days of the drug, 30 days later, and ultimately comparing that to survival. So I'd like to acknowledge people that were involved in this. At the Koch Institute, a number of people, including folks from our imaging and histology core facilities. I see Kathy, Charlene, shout out to you guys for your help in the study. Then a number of people from my institution at Israel were involved, and other places as well, from those sources. So I'm happy to end there. I'll turn it over to Adam, and I think we'll leave questions today.
Sure, you're going to figure it out. This is Adam, our data scientist. I want to say, isn't it 10 times more interesting if you see it all put together, right? You know? Yeah. Instead of just a sample. You good?
All right. Hey, everybody. I'm Adam Patch. I'm a data scientist at the Koch Institute, specifically stationed in preclinical imaging and testing. So right next door to Histology.
Yes.
And I'm going to talk to you today about knowledge graphs. So apologies if it's a little too nerdy, but I'm not a computer and data scientist. I think one thing I really like about being in core facilities is the opportunity to collaborate across a lot of different points of view. And I'd like to show you how this kind of technology can hopefully help with that. But starting with a question, because we're still very much in experimental phase with this. So the last few months I've been testing out a number of use cases with this. It was exciting to get invited to talk. I know Kathy's spoken with my director, Virginia, a number of times about this work, and I think there's a lot of benefit that we can see potentially on the course. So the end state is something I just started working with recently. And here we go. So the big question here is, can scientific knowledge be stored and retrieved digitally? So here's the deep dive into that. So an overview of my talk, I'm going to give kind of a product explanation for why I'm looking at this, so some context needed and the vision we have. And then dig into advanced data a little bit, look at this experiment and how we can represent that. This really wild image on the right is a portrayal or representation of the advanced data with the MRIs and body masses of the mice and a number of other entities. I'll get into that. And then contextualizing this in other ontologies, so other frameworks, other ways of describing this. I'll walk through a number of those. Dan showed you the EDA just then. Jeffrey is here. Our microscopy board has been working with open microscopy environment. The other committee of animal care, PubMed. I'll try to show these quickly today. But so first, I'm going to just give context to this. So I joined the Koch about a year and a half ago and started thinking about some big questions for data in our core, and then also thinking about how we can help, connect it in between cores. So you saw in Dan's talk, there's a lot of that in a project like his. So the big context that we've all been thinking about, I think at least in the data side of things, is this data management and sharing plan from NIH. You see on the website, it started in January. I know people are up in arms about this. Actually, this is what connected me with AVRF in the first place. There have been some great webinars about this. Then just within that and beyond that, the fair data principles. So we should be making data more findable, accessible, interoperable, reusable, and in my point of view, also computable. Then our facility works with animals, so we have to deal with the 3 R's of animal research. And I think data has a lot of opportunity to break down the silos that prevent us from doing things like producing, refining, and replacing experiments. If we have data already, there might be a way to avoid rerunning an experiment that isn't necessary. And then, I think of core facilities as shared labs, shared resources, and knowledge, and I think data plays a role in all. This image is a goofy data science image. Okay, so these are all nice things to say, but will the data share itself? And I think this is something data scientists confront a lot, is we're ready for analysis, but actually we need to clean up the data. And in science, data is always messy, especially in cores where we have shared instruments, but not necessarily shared storage or things like metadata that might give context to the information that we're trying to keep track of. So I'm going to dig into some of the sort of harder constraints that we face. Like NIH, as a core, we don't need to look at that, but for the sake of good science, we really do need to make more reproducible research. And I think this is a problem in animal research. This isn't my background. I come from physics, so I'm learning about this as we go. But I believe a lot in quantitative rigor, and I think things like computation, segmentation, and avionics are all part of the data scientist's job in this environment. We also need to accommodate for a lot of complications. So here on the side here, you see all of the instruments in the facility I work in. I'm getting some of the same funny imaging feedback in out here. But you can see most of them. That's the scary one. But yeah, basically, they're big files, so where do we store them? Complex morphologies, how we match between different images. We're looking at mouse body, which literally changes when you move it from modality to modality. Then there are also unique workflows where there's proprietary instrumentation, proprietary software. In advanced data set, for example, the MRI data files changed because it went over the time our vendor changed it. Then there's compliance and oversight. Consistent. Then, okay, you saw the base form here, but there's this desire, I think, in a core facility to recall failure. This is something that I've heard about in talking to people in my facility. That we know it's going to fail. There might be a publication out there that claims it works, but we've seen that when you combine these particular factors in an experiment or in a study, that we know it's not going to work. We've seen it in our facility already. And then there's this even harsher reality that sometimes audits happen. So something's been published. We need to dig back into data that's been produced in our facility. And then, I would like to be doing things that are state of the art in data. So I'm putting this kind of fun model up. It came out in the last couple months from Meta, Facebook, but it's helping. The next slide is really goofy, but I like bringing up memes. I think it can be fun. And this particular one came up in reading about databases. So there's this video. Oh no, we can't read this. So it's like an argument in the late 80s over SQL servers. Real nerdy, but it's Bill Gates, who had Esper, who I had to look up. And they're arguing about what's the best thing about a SQL server. So just so people know, a SQL server is where you store the data, you can query it, and pull out information. It's all in tables that are related to each other. And they're really excited about this back then because, Ed Esper wants to use one kind of software, DB4, to share data. And then Bill Gates wants to use Excel, which is still widely used. And so, they're arguing about which one's better, blah, blah, blah. But then they realize, wait, we're using the same data here. And that's actually a really big thing that I wish we could start doing more and more in core facilities, using the same data. so that we can not just check redundancy with experiments, but with analyses too. And so thinking about this like it's the same data, how can we envision some new approaches to using data in imaging research? So I'll just kind of really briefly unpack some big terms from data science that I get excited about data science. I changed careers from physics into data science because we're in an era when data science tools are just exploding. There's so much that you can do really out of the box with many different models, and there's a lot that's resulted from a few waves of data stuff. And big data was one of these things that people talk about quite a lot. So this focuses on things like having good databases and data structures. And the real outcome of that is having good search and storage at scale when you're in a lot of data. But then data scientists usually are trying to approach this saying, okay, I want to explore the data, find new insights, do more modeling. Whew. Sorry guys. So that says Julia, Python, and R. Anyway, you don't, again, maybe just jargon, but the idea is that there are these tools that are widely available right now, and we can put in the hands of researchers that allow for machine learning and even automation of rerunning studies, again, at the scale that we need. But what I'm thinking about a lot right now is how enterprise data approaches can help us. So, there's been a lot of work in more recent years to try and make more value from data in industry. And that really means making more cross-functional tools, no-code tools, and doing things that rely on natural language that domain experts might use, like scientists don't work with. I will admit, I don't know most of what gamma is, and that's actually part of what I like to bring to this, because I think there's an opportunity for us to make sure that we're explaining these things well, and that there's a lot that we can do to translate between the different ontological approaches to this kind of work. And the other big thing about this is curating data sets well. So, with good curated data sets and explainable models, we can do rapid drug development and delivery. So, I'm thinking of this as something like data management and channel scale. So, again, my background is in physics. That's why I put that down at that time, because there's a lot more to science than data and knowledge, but I'm focused on those two here. And so we think of data in the lab. We're dealing with things like spreadsheets and images, things like a specific organ, a specific compound, specific amounts of a drug, duration of time. That, to make it into knowledge, becomes a statement, right? Well, we're going to say something like particles smaller than the size of X accumulated in organ Y after Z hours post-administration. And then compounds with property A reduce their tumor size at just B. And then we use that information in publications, pull that back, start collecting more data. And so this approach, technologically, that I'm really interested in is one of knowledge graphs. Neo4j is the platform that I'm using, and this comes from some of their literature about how knowledge graphs work. I think it's a nice diagram because it shows what data looks like when it's not connected to other data. When you add relationships between data, you get what we call a graph. So we have all of these nodes. Maybe this is something like the internet, where there are websites with URLs. And then a URL might have a number of links to other URLs. But without knowing what it is in the actual website that's connecting the two, we don't really know much about these connections. So adding semantics to it, or language, allows us to actually structure the way these things are connected to each other. And they call this dynamic context and deep dynamic context. To me, what's really important about this is that we have a framework to understand complex data. And this is very flexible. A real-world example of this that has helped me explain this more than the lab examples to my colleagues is the Wikidata knowledge graph. So I think people use Wikipedia all the time, but maybe don't realize that there's a knowledge graph underneath it. And so they have a way to query this knowledge graph that's open online, and I can go and Google search for a query that gives me, say, all the hospitals in the world that they know about that are in the knowledge graph. So I can copy that query into here, run that query, and look, it's like Google Maps. I get a map of all the hospitals around the world. But then what if I want to zoom in on that map? Like I live here in Boston, so okay, if I want to find a hospital that I know is like maybe Mass General, I know where it is because I've walked around, but I wouldn't, I guess I can Google search it. But in this case, I'm trying to show you how I can know where it is just spatially. And you see here, I can zoom in, find Mass General, click on the link in there, and then get some more information about it. And not only that, it's probably not about the read, but I can see how Mass General is referred to in a number of different languages. And this is the sort of ontological thinking when we do multimodal work or collaborative work that I think can be really helpful. So now back to the next experiment. Thanks for bearing with me for all of the explanation of why I'm interested in knowledge graphs. But I want to show you just quickly. So, sorry, sounds louder. So Dan's subject and imaging data here. This is what it looks like when I get a spreadsheet from Dan. Hopefully this is too small for you to read, but there's so much more in this. I don't know if you can see the scroll bars, but Dan has packed lots of very valuable information here. So like, he was showing you the results of this, he was showing you how we thought about coming up with this, which by the way have a lot of arrows, nodes, and edges. Anyway, but then what do I get with this one? I look at it, I see these 8 cohorts that he's collected from 2020 until the present. So there are multiple data sources here, and then notes on each subject. And then he's conducted a number of treatments, so radiation treatments, pharmaceutical treatments, and then controls their mouth. And he has a number of MRI time points, so these vary depending on the cohorts, but he saved the files in a nicely managed file structure. And so when I go to reach out for those files, I have all the information that I need based on the subject name and the date to collect the DCM files automatically. Dan has a question, could the tumor shrink? And I want to be able to answer that question quickly. So this is not quick. I'm going to actually dig into building a data structure so that we can answer it quickly. But here's how I query advanced data. So there are a number of entities in this data that I can group this into. So we see things like an experiment, a cohort, a subject, pharmaceutical interventions, radiation interventions, tumor masses, body masses, a number of details about imaging. And basically I put these into a schema where I can say that an experiment included a pharmacological intervention or pharmaceutical intervention. An experiment included radiation intervention, and then I can point to which of the subjects that intervention happened to. And then within each of these, there's more information and properties. That's how I can then take this data and then type it into a knowledge graph. And so this is an example of one way to view Dan's data in a knowledge graph. Here, what you'll see is something, again, that's not discernible to just look at, except maybe to get an abstract notion of what all he's done, what he's collected, and how densely populated it is. But then you can also see in the upper right the types of nodes that are in there, and they're color-coded. So you can see the experiment that is at the center of this, then also the different cohorts, there are 8 of them, that listed out up there. And then the different variations on pharmaceutical interventions, body masses, how many measurements he's made, so on and so forth. So this kind of thing can actually fully summarize his data set as far as I need to know, because I know how many things are there, how they're related, and I can pass this along to someone. In fact, I passed it along to my director right when I made this, Virginia, over Slack, and in 5 minutes she got back to me and said, okay, let me guess. What is this? And then she took a guess, and we basically, communicated in one graphic what Dan had done. He had 8 cohorts, he collected all these body masses, he collected images, and he had done imaging just with one modality. So she got all of that just from this. I like that. That's great. But if we want to dig into this deeper, I can write a different query and just pull out a few of these entities. Like, if we just want to know the subjects and body masses, etc., then I can start breaking this down and picking it apart. So I could show this instead of the entire thing. And then I can even take this a step further and produce a table from this. So a table that maybe, does a little bit of math. So that when I'm sorting everything here by subject name, by date, and then the tumor mass and the body mass, and then we can see the ratio between the two. And this kind of thing could be useful for a downstream analysis. And so, analyzing, this should say analyzing with metadata, but the idea here is that fair data is curated data. And so you know, in image analysis, I think a typical approach as needs be is a one-off, basically, even if it's with machine learning. Quite often, there's a need to train on a data set that is very relevant to what we're trying to segment. So the more relevant that data is, the better. Here I'm showing a few slices from Dan's data set, not effectively segmented, except that I've been able to automatically draw, you know, contours around the actual body mass to get an estimate of the total body volume. When I do something like this, though, you know, there are a lot of questions to ask, like what about the specific tumor type, what about specific organs, what about how this was prepared? And so this big goal that we have here is to overcome challenges without reinventing the wheel for these things. And so to do this kind of analysis in a more fair way, a verified way, you know, my approach is to use a knowledge graph. And so if we use a knowledge graph, we can make data sets like this findable. Then, you know, if we find the data set based on its metadata, it's accessible if we can get it from storage. Then we can load that into some software here. I'm showing the Jupyter Notebook, which is a data science like passport throughout the world. You can use with almost any data because Python tools are ubiquitous, along with Julia and R. And I think by being able to access it from a point like this, we are able to a lot of research. And then things like documenting and storing analysis, if we do it in this way, then there's a way to not only recall the analysis or the data, the results of the analysis, but the actual code behind the analysis, which makes it reusable. And to do this in a way where we can build these queries right into the code makes it computable. So in principle, someone with access to a database like our knowledge graph could rerun a notebook like this and reproduce the same exact analysis on a new data set that is similar enough and tested. So those are some ways we can apply this, you know, specifically to Dan's data, but then also, like, how can we use this to bridge between different contexts? I think there are a lot of really great digital tools available now. I mentioned some at the beginning of the talk, but how do we make it so that our data actually connects to these things, and we're not manually trying to connect everything all the time? So, like, one really great tool that I found for this is through PubMed. So PubMed has these e-utilities that are connected to the BioPython package, and I can basically write a query on the entire PubMed database from BioPython. So this allows for search and utilization of the indexing that PubMed does, and the framework that PubMed uses, so that we can recontextualize that into our set at the Code Institute, specifically in imaging and testing. Then, you know, the cool thing about PubMed, though, is that underlying this is an actual ontology that they've developed called the medical subject heading ontology. And so this is the only video I have in here. I will try to run it, but this is just an example of what I can do with the PubMed ontology in the platform that I'm using. So this is an example here. You'll see the canvas, and now I open this sidebar where I can store queries that I can share with a user that point to specific parts of this knowledge graph. So then when the user goes in, they can just search for that query and run it. In this case, it's a tree showing anatomy descriptors. And it takes a second to load, but once it loads, you'll see this tree that I can put into a hierarchical format. And we can zoom in and start exploring what ways the mesh headings actually can be used to describe these different parts of anatomy. And so using something like this, I think, becomes really powerful because it allows us to start categorizing the things that we want to talk about and store as knowledge in a database for our use internally. I won't go through this whole video. But then, okay, so I pointed at Jeffrey before, I'm going to point at him again. He's been working on setting up an Amaro server, and I'm really excited about this because, like the Wikidata example, the Wikipedia, you know, one of the great things about Wikipedia is that it allows many people to edit the same information and to view it as well. And having tools like Amaro, which are designed basically to give a platform for viewing, seeing and editing metadata, and even working with images is really helpful. So in thinking about how we can make something that allows us to interface with that nicely, Omera is especially nice because it has the ORM data model, which already does a lot of work to convert things through bioformats, through OME files that have built-in metadata, and there's an active community, developing and working on this. So, the thing that I like about this is that they have an XML ontology, basically, an XML with a schema that I can use and build into an ontology. And this kind of work makes it so that someone like me can grab this and maintain a code library that's going to actually connect with information on a local resource like that. Oh, and well, you can't read this at all, I really apologize. There's too much to say here, but this is an example of the image data resource, the IVR. These basic names sometimes screw me up. You can see on the left a list of all the images in this particular data set. And on the right, you can see how metadata about this experiment can be stored. And then if you were to dig into each of the images, there's metadata about each of those that can be stored and edited in here. As Dan mentioned, there's also this tool that we've been trying to promote using, which is the EDA tool from NC3Rs in the UK. Now, this is recommended by NIH for researchers doing animal research to collect information about a study, and it's really great because it's so visual. It's also really great because it has tools for analyzing things like doing power analysis of your study. But I have to say, seeing Dan's experiment in there, I understand quite a lot more about it, and I'm excited about it because I can actually I showed this drop-down menu here because there's a way to export these files. They're framed in terms of nodes and relationships, which means I can write a script in an afternoon that pulls all these files in to a database like ours. So once again, there's a metadata editor. This one's especially nice because it's in a graph structure. It's NIH recommended. We'll see if people adopt it, I guess, but I love that I can export it. And this is an example of one of their templates. I wanted to put Dan's data in here, but I didn't get to it yet. So this is one of their templates in my knowledge graph, and I have 5 of these and can search through them. We've also had some of our users start submitting these to us as well. Then there are these other digital resources that are really valuable to us. So at MIT, the Committee on Animal Care, which I know is IACUC and many other places, is finally digitizing protocols. So storing these in a database for their use is great, but what we would really like to know is when certain factors of these things are going to impact the way we have to work. And this is something, it's been stored in these 60 to 120 page long PDF documents that have to be referred to all the time, stored in our own Dropbox or somewhere else, communicated about. It's very complicated. And I think that we're getting really close to a point where we can actually just send these details through an API, again, automated. And then the last thing I thought would be fun to show is that I made a data graph of our data management plan. So in our facility, we don't need to make a data management plan like many of the labs we serve do for NIH, but we've been working toward developing one so that it's easy for us to pass along information to our users. And this is an example of one that I put together based on all the terms that we came up with as a team. And this is another one of the Neo4j's tools where we just go into a web browser, it connects to my Google Drive, and we can just draw this up like a whiteboard. So that's how we put this together. So I'm not endorsing Neo4j, but I'll just say a couple things about them that has made them a really good platform. So they're an active thriving community, much like Omera. They have conferences annually. We have a free community edition. And then the language OpenCipher is open source, so it's really nice to work with. It's educator friendly. Right now we're using a free enterprise edition for educators, which has been great. But that means it's also enterprise ready, so it's scalable, it's lightweight. It has a full toolkit for setting permissions and federating access. And it's really flexible and portable. I know if anybody here has ever worked with sort of ontologies of the 2010s, things like RDF and OWL language, then you know that that's not really very flexible. It's actually quite difficult to work with. I think a lot of people have kind of given up on ontologies as a result. I think a new platform like this is something I would not really try out until I saw how accessible and no-code friendly it's become. So I think it's worth pointing out that there's a really easy way to adapt schema in this new platform. And the coolest thing about it is that it maps to these other standards. So I can develop a NIST and then put it in RDF or something else more complicated if we want to put something in production. Oh yeah, include spreadsheets. That's very important. So, like Dan said, this is potential. We're still experimenting with this. So there are a lot of next steps. And actually, one of them is that we're going to use some of these use cases to go through some focus groups with our users and other core staff and see what's most important. Because I don't know if you can tell, I'm scatterbrained about this. There are like so many things to connect to and so many use cases. What do we do? So we're going to reach out and get feedback on that. The fun thing for me is building these ETL pipelines where we extract, transform, and load data from all these different places. So that is making those production ready is sort of fun step. And then, getting this database communicating with the whole data ecosystem network AI is something that we're really excited about, and why I think it's so important to use something so flexible and modular. So anyway, I'll kind of leave it at that. Knowledge graphs really have a lot of potential to enrich scientific data with meaning, help us centralize this scientific data, help federate access to improve data sharing, and connect new tools and domains for multimodal research. So, bolsters these things we hear about and help against good science. Thanks.
Are there any questions?
Yeah, I can hear you.
Adam, so you talked about the potential of natural language output.
Yeah.
What about the potential of natural language input to get researchers to use it, to generate these ontologies a lot more and eventually hopefully voice activated natural language input? So you can just describe your experiment and have it generate like that.
Yeah, that's something I've been actually playing around with a little bit. I don't have anything like really meaningful to say about it, except that I can go to ChatGPT and ask it to make me a knowledge graph. And what it does, I'm definitely not here to endorse ChatGPT as a final solution because it always gives me bad stuff, but in a great format. So it's like having an assistant who knows how to perfectly format things. And then I can say, hey, can you rename those variables? And then I do it. But I have to always go back through and edit some things to make sure it makes sense. So there's that. But then also the interface with query library like this, I guess that's not what you're asking about. One thing that I've done that is too creepy to put up here, but I'm a data scientist, so you guys all know I'm creepy, I guess. So our website has a people page that lists all of the faculty. And so as a test case, I went to the Lambda Lab, downloaded the research summary, which I can do from the command line, and then asked ChatGPT to make a knowledge graph of that. And it would be cool to show you that right now because it did a pretty good job, just from the entities in there. Again, I rearranged it so it fit what we wanted. But yeah, I think there's a lot of potential for that. But human and always.
How do knowledge graphs interact with bias packets, with bias statistics?
That's a good question. So, I think of it fundamentally as a data structure where we can store information, and ideally information that is a little more complicated than a table. So, if you want to store something like a lot of results that are all the same, a knowledge graph is probably best to just point to that as a file. But in this case, for example, with Dan's data, I can do some basic statistics just on the entities that are in there, like the log analysis. I can look at the dates. I can reproduce the survival plot that Dan showed. the cool thing about something like this, I guess, that moves beyond that, like just having a spreadsheet, is that we can have that spreadsheet, put it away, and then recall it when we want it, based on the context. So if you have a number of spreadsheets that correspond to different studies, like let's say you've accumulated a lot of data, then you might want to do statistics on a very particular combination of features in that data. And you could locate data that correspond to that using the trap.
So I'll say, just from the other side of things, there are lots of biostatistics programs out there. I think you learned probably from my presentation, I like to use Prism. It's one of the ones that I think a lot of people are using now. So I'm just thinking about the question. It would be interesting to figure out a way that we could get knowledge graphs about the Prism and vice versa. We could start to analyze things in a way that we'd like to do.
Great presentation, thank you. So I really appreciate this idea of finding new ways to display and project the data to understand it. My question is actually a few steps back, thinking about multimodal imaging, the input data and the co-registration of the data that's coming across different modalities and the normalization potentially of that. So we have very different input, output, sensitivities. What are you thinking in terms of that aspect of what we do to get high fidelity data displayed on the back end, that we're actively recapitulating what's happening at each step and again co-registering that data?
Yeah. I mean, I think you're kind of picking out all the pieces of that that we need, right? So, to be able to co-register, I guess I showed how I can store those files, but what we can also do is store coordinates for those files. So, this is why I like this Wikidata map example, because you imagine overlays, in 2D like that. We might want to do something in 3D that gets one coordinate more complicated. But, I think what still is something I haven't dealt with is how we deal with the different morphologies once we get to that stage. I imagine though the data structure like this, as you're going through a project, if there are important points to keep in mind about it, you can store that in the knowledge graph relevant to that project. So then upon recalling that, there's an extra note that says, oh, to co-register, we needed to move this or that image, this amount, or we needed to shrink or grow, or do other kinds of radiometric tricks with this to make sure it works.
Hi. Oh, I'm sorry.
Yeah, I'll just mention the last question that I was asked. It's a challenge dealing with these imaging studies. I had 100 subjects in the study that I showed you, times 3 MRIs. It's challenging, and this is something I'm pushing at because right now what I have to do is I have to manually go through each of these, and I have to conjure out tumors. It's a lot of work. I'm hoping that we can get better.
We have a question from Virtual Land. This is from IKEY. How is a knowledge graph different to the structure of a relational database?
That's a great question. I have this kind of, I don't know if it's a snarky response, but it's like a hyper-relational database is how we think of it. The sort of trap of relational databases with complex data is that oftentimes when you build a table, it has a use case in mind. But then in that table, you have a number of columns, hopefully unique identifiers so you can relate it to that entity in another table. But the table itself isn't fundamentally descriptive. So to go from a table to a knowledge graph, you have to add semantic meaning to it still. Like you have to say, how are these columns related to each other? And how does that fit into an entity? Which properties do we want to include there or elsewhere? How do we relate them? That does take a little bit more effort, but it can also be so specific to the use case that you're talking about. If you have a relational database, again, these are incredibly useful for consistent data. So there are lots of use cases where you definitely still want to use relational databases, but if we need to include some new fact for this experiment that just doesn't fit into a table, what are we going to do? Rebuild the table or make a new table? I have limited experience in industry, but the year that I spent as a data scientist outside of academia was with a company that had a legacy database, lots of data. It was a social media site for healthcare, and it was just tremendously large, like what we would look at as newcomers. how do you go through this kind of ecosystem of data that engineers of the past built? If there's not documentation, it's almost meaningless sometimes, even if you can go through and read the column headers, you know the table names. Building queries is more complicated too, and those are the two main things that I think.
Thanks, that was gorgeous work, and I love the systems you put in place, but sort of in the same way, the plan never survives contact with the enemy. Stuff like this often never survives contact with the user. How do we incentivize people to actually fill this in, especially when there isn't sort of professional data scientists on staff?
Yeah, these are kind of maybe even open questions, so I think this is, again, the reason I try to think of like a product in mind. Like, because to have a user interface, you need to think about what that experience is like. And so having tools that are more accessible, like, okay, we have this database. I've showed it to my colleagues on my team, and only a couple of them actually want to like get their hands dirty with it. But if we can connect it to things that people use, like Omero or, I'm blanking right now, but there are a number of ways I think we can make access points to it. And then for users who are interested, I think it's a great opportunity to learn because, look, Neo4j, for example, at the last conference, they had the director of data from Pfizer talking about how they use this to deliver COVID vaccines. So they get a call and they want to know if that vaccine was delivered, when it would be delivered, and the executives can use this. So I think that's a real testament to the power of using something like this, but TV. Yeah, probably.
Yeah, again, I'll give the other perspective from the person who uses these things. I think what's critical is to find platforms that are user friendly. But Bill Gates was right. Excel's a great platform. I'm an Excel guy, but I think time has come to move on to things that are able to integrate different platforms better than Excel spreadsheets, which I'm still working. And the EA website, I'll just give you an example. This was something that that second to last slide that I showed you, which was the flow chart of the trial, that was something I generated over the weekend while taking care of 2 young kids at home. I don't like to learn new programs, but it was really easy. So it was an easy platform to work with, and I think that's critical. When you're interfacing with researchers, you have to give them something that they're able to work with quickly and easily.
Anybody else?